Hello everybody, welcome to Ms. McGuire online video lecture, and today we are covering gene mutations. This video lecture will be divided into two parts. This is part one of the chapter. So let's go ahead and begin. So learning objectives for this chapter include the following. So after completing this chapter, you will be able to distinguish between mutation and mutant, define polymorphism, distinguish between a germline and thematic mutations, describe mutations in the genes that encode beta-globin and collagen, explain how mutations arise spontaneously and how they may be induced, describe the two types of single base mutations, explain the consequences of a splice site mutation, discuss mutations that add, remove, or move DNA nucleotides, describe how pseudogenes and transposons can cause mutations, list the types of damage that DNA repair mechanisms fix, and describe the types of DNA repair. The nature of gene variants. The term mutation and polymorphism each denote a genetic change from the wild type. So the type of organism, its phenotype, how organisms look like, that is most common in nature called wild type. So any time when we have phenotype that is different from this most common form, we use term mutation or polymorphism. Mutation is a change in a DNA sequence, and mutation is rare in the population and typically affects the phenotype. Polymorphism simply means many forms. It is a change in a gene that is less rare than a mutation and more prevalent in a population. The nature of the variants continue. The effect of mutation vary. We can have loss of function mutations, we call them recessive, or gain of function mutations, dominant mutation. Term mutant refers to phenotype. Usually connotes an abnormal or unusual or even uncommon variant that is nevertheless is normal. So when we have mutation that changes the phenotype, right, it doesn't necessarily something abnormal. It can be actually just another variation within population. So think about eye color. We have people with blue eye color, with brown, with green. So neither one is, abnormal. It's just normal polymorphism, normal variation within human population. Some might be more common, some might be less common, some can be maybe unusual, but still considered to be normal. Mutations are important to evolution. Our evolutionary relatedness to other species allow us to study many mutations in non-human species. So if we divide mutation in two large groups, we can define them as germline mutation and somatic mutation. Now, germs means human gametes, ova and sperm, so eggs and sperm. Germline mutation happens when change occurs during DNA replication that precedes meiosis, and it's transmitted to the next generation of individuals. So if we have, for example, mutation in an egg or a sperm, and if this egg or sperm were used in fertilization, then all these cells of the organism that arise from the cells that had a mutation, all the cells will have it, right? Because we start as egg, right, and sperm. So we start with one cell that's called zygote. So if this zygote has a mutation that was carried by egg or sperm, right, then the entire organism will inherit this mutation. So every single cell will have this mutation. This is what germline mutation. Somatic mutations, however, happen during DNA replication before mitotic cell division. So that's already when eggs and sperms are formed, because they form by meiosis. So if egg and sperm doesn't have a mutation, and mutation happens later during embryonic development or even in an adult organism, right, then it only will affect the cells. Well, if it's during the embryonic development, it will affect the cells that descend from a changed cell. And also, even in an adult organism, for example, if you have cell and it has a mutation and cell became cancerous cell, then when this cell divide by mitosis, then all the cells that descend from this cells that had a mutation will have it. Now, if we're talking about still embryonic development, then person with somatic mutation has somatic mosaicism. And I will show you on the next slide what it means. So what we want to take away from this slide, what we want to remember, from this slide, that mutation can be germline mutations that happen in DNA before meiosis, or somatic mutations that happen in DNA before mitotic cell division. So here's an example of somatic mutation. A dog with genotype little e, so this is homozygous recessive in a specific gene called AMC1R. It's a color gene. So if a dog has a mutation, then it would be golden or cream colored, like this one. Whether a dog whose genotype is big E, little e, is black or chocolate, right? But now if you look at this dog, so how happens that, it's gold and just a part of it is chocolate? Now, because it's a somatic mutation that happened before the birth, and it caused a pigment patch. All right, and if this doesn't make sense, let's look at this picture first, and then we go to a previous slide. So here's our DNA, right? And so imagine that this is zygote, the first cell, and then this cell start dividing. So here we have a ball of cells, and just one cell spontaneously gets a mutation, right? Now, all the cells that will arise from this cell with a mutation now have it, but the rest of the cells don't. That's why the organism after birth, cells with the mutation can, either grow faster, as in this example, and show body asymmetry, right? But it's not entire organism that is affected, but just a part of the organism that, was developed from the cells that had mutation. So now that's our previous slide. When somatic cells mutate in an individual, not all the cells in that individual have that mutation, because not all the cells are affected by the mutation. The individual has somatic mosaicism. Patterns arise as affected cells migrate during development. This has happened in Proteus syndrome. The few hundred patients have overgrowth of skin and bone, but only in the body parts that bear a mutation in a gene AKT1. Right, so, and that diagrams that I showed you related to this mutation in gene AKT1, right? So it's shown over here. Now, the boy over here has Proteus syndrome. Now, it's a rare genetic condition that causes excessive growth of bones, skins, organs, and/or tissues. This disease causes part of the body to grow out of proportion with the rest of it. The overgrowth is usually asymmetrical, meaning it affects the right and left sides of your body differently. And in this boy, you can see on the left side, his fingers are much larger. Also on the right side, his spine and bones of the right side are larger as well, that give asymmetry to the body. Now, why did it happen? Why not all of his bones and organs grow fast? Because that's a somatic mutation. It happens after fertilization, right? So it was not an egg, it was not a sperm that was carrying this mutation, but during embryonic development, some cells develop mutation, then they migrated to a different part of the body and they promote the growth of this part of the body. And that's what somatic mosaicism is. Here we will look at mutations that alter protein. Identifying how a mutation causes symptoms has clinical application and also reveals the working of biology. Examples of mutations that cause disease are shown in a mutation of beta globin gene and collagen gene. Now, beta globin is the protein that is important in hemoglobin. Now, you have red blood cells. Inside the red blood cells, you have proteins called hemoglobin, and hemoglobin carries oxygen through your body. Hemoglobin is made of alpha globin and beta globin. So when beta globin gene that produce beta globin is affected, it affects the shape of the cells. So here's one disease called sickle cell disease. It's a first inherited illness understood at the molecular level, results from a single DNA base change in the beta globin gene, which replaces glutamic acid with valine. So let's go to the next slide first and see what it means. So here's the DNA, and at a particular part of the DNA, we have sequence C, T, and C. And when we make RNA from a DNA, remember, RNA is built following the complementary base pairing. So C always pairs with G, and T always pairs with A, and C with G, right? So here we need GAC if it's a normal gene. Now, GAC codes for amino acids glutamine. Right, so that's going to be a normal protein, normal beta protein, and this is how those protein looks. There is no aggregation of hemoglobin molecules because beta protein, as we already mentioned, is part of hemoglobin. Now, this is what happens if a person has a mutation in his or her DNA. So instead of CTC, the person has CAC. And when RNA is made, A pairs with U. So now instead of GAG, we have GUG. And unfortunately, GUG codes for different amino acid, valine. And when this valine is added to beta globin, then hemoglobin that is made of beta globin and alpha globin will have abnormal properties. It will cause abnormal aggregation of these hemoglobin molecules. So instead of being like a free hemoglobin molecule, it forms this aggregation and it changes shape of the cells because this hemoglobin is inside red blood cells, right? So now we go back to our slide. Hemoglobin molecules aggregate into long curved rods that deform the red blood cells. Sickled RBCs or red blood cells lodge in a narrow blood vessels, cutting off local blood supplies, causes great pain in the blocked body parts. Sickled RBCs are destroyed in the liver and spleen, causes anemia. So now the person doesn't have enough red blood cells. And anemia is low oxygen delivery, right? So this is just a simple base substitution. So we substituted one nucleotide with another nucleotide in DNA, it's affected RNA, and it's affected protein at the end. And when protein has alternative shape, it can aggregate in this example, and it will cause the cells, red blood cells, right? This is normal red blood cells. So here's normal hemoglobin inside, but those are sickle cells. Those sickle cells has this type of hemoglobin inside, and those cells are removed from circulation. They can be stuck inside blood vessels, and this causes symptoms of sickle cell disease. Another disease, thalassemia, caused by another beta hemoglobin mutation. So the same protein, beta globin, that part of hemoglobin, but a different mutation. And this mutation results in too few beta globin chains. And hemoglobin is made of beta globin and alpha globin. So if you have mutation and we don't have enough beta globin, then we have excess amount of alpha globin. And this excess amount prevents formation of hemoglobin molecules. Then we have a free ion that is not attached to globin that slowly damages heart, liver, and endocrine glands. Thalassemia minor develops when a person are heterozygous. That means, well, you have 2 chromosomes, right? You always have pair of chromosomes. If in one chromosome you have mutation in this beta hemoglobin gene and another chromosome doesn't have mutation, then a person has like one functioning gene and one gene that is not functioning. So it's a heterozygous. So it will develop minor symptoms. Or thalassemia major, when we have homozygous for mutation, it's a more severe condition. So you can see the blood smear over here and see how some blood cells look, misshaped, like here's some pieces of it, or this one. So that's another genetic disorder that is caused by mutation in beta hemoglobin gene. Another example how mutation can affect our function is collagen mutation. Now, collagen is a protein. It's a major component of connective tissue. And connective tissue make bones, cartilage, skin, ligaments. Ligaments attach bones to bones, tendons attaches muscle to bones, and tooth, dentin. So dentin is chemicals that make your teeth. So that collagen is a major component of all these structures and organs. So you can imagine if collagen, and it's a protein, if it's not synthesized correctly, all of these organs and structures will be affected. We have more than 35 collagen genes, encoded more than 20 types of collagen molecules. Plus, we have genes that affect collagen as well. So they're not collagen genes, but they still affect the structure of collagen. So mutation in these genes, not surprisingly, lead to a variety of medical problems. So here, just a short table of some collagen disorders, and we're not gonna look at all of them. We will look at the osteogenesis imperfecta type 1 and Ehlers-Danlos syndrome. Actually, I think we're only gonna look at Ehlers-Danlos syndrome, but osteogenesis imperfecta is just a cool disorder. We call it blue sclera disorder. The person with this type of condition, the white part of the eye that called sclera, it is actually looks blue. That's why they call it blue sclera. So they have like not the so your eyes, you have color, right? You have eye color. You might have blue eyes, but that's the color of the iris. But the white part that is sclera, if you don't have genetic condition, it's white. Those people have the sclera that is blue. Okay, but you can appreciate over here how many different mutations we have that affect the structure of collagen. So here's the structure of functional collagen molecule. So it's it's a triple helix, right? So we have three different strands, two the same, right? Shown here in blue, and one different that is red. So this is the longer precursor, procollagen, and then this procollagen needs to be trimmed to form a collagen. So this parts need to be cut off to make a functional collagen. Hold on, yes. This is next one. Let's see. Right. So okay. So it's not described over here, but here's our here's our Ehlers-Danlos syndrome. This mutation prevents procollagen chains from being cut. And then collagen molecules cannot be assembled and skin became too stretchy. So this is one example. So if we have a procollagen, so if you have procollagen over here, and we need a special mechanism to cut off these terminal peptides to make collagen strong and have a great tensile strength. So collagen actually prevents your skin from being too stretchy. But if you don't have functional collagen because of the mutation in gene, then your skin became very stretchy, as shown on this picture here, right? Okay. So what type of mutations we have? What are the causes of mutation? So far, we described the, so let's just go back and go back to see what we covered. We said that mutation is a change in DNA that affects phenotype. It's not common. Polymorphism is just different forms within a population, and it's more common. We just also said the mutant is a phenotype that refer from mutation. We mentioned that mutations, not necessarily harmful. Sometimes they're just normal, that give us varieties. That's why we all different. Different blood type, different hair color, eye color, it all arises from mutations. We also said that mutation can be somatic or germline mutations. So somatic, they affect the cells after fertilization, and germline mutation affects the formation of gametes, eggs and sperm. We described what somatic mosaicism means, right? And we give two examples of mutations, a mutation of beta globin and collagen, and how mutations in the genes that code for those proteins result in presented phenotypes that we described, like sickle cell anemia, thalassemia, right? That's a mutation of the globin molecule, beta globin molecule, or how mutation in collagen, and collagen is so essential for many organs, can cause different disorders as well. But what causes mutations, right? So I'm going to cover a couple more slides, and then I leave the rest of this chapter for part 2. Now, what cause of mutation? So mutation can occur spontaneously or by exposure to a chemical or radiation. An agent that causes mutation is called mutagen. So we can have spontaneous mutations, de novo or new mutations that are not caused by exposure to known mutagen, and result from errors in DNA replication. So spontaneous mutation mechanism are shown here in this diagram. So here we have a parental DNA shown in dark blue and a new DNA in a light blue. So for cell to divide into two daughter cells, first they need to replicate their DNA. So you can pass, chromosome equally between two daughter cells, because all human daughter cells, if it's not eggs and sperm, they must have 46 chromosomes. So that means DNA need to be replicated. So in this example, here's our parental DNA, and it has nucleotides C, A, and G. Remember, C pairs with G, and A supposed to pairs with T. But in this example, the wrong nucleotide was added. So it has a G now. All right, so, maybe one strand was replicated with a mutation and one without. So this one, they show that if we have mismatch in one DNA, right? So normal DNA has sequence CAAG. Now we replicated DNA and we made a mistake over here. Now when this DNA replicated again, because cells continue to divide. So when this DNA replicated again, the strand of the parental DNA will be unaltered. So there is, because we still have CAAG, and the second time we didn't make mistake, but our first mismatch strand, that is GTC, now will affect the second strand, right? So the DNA will have a mutation, right? So this is our, we replicated DNA one time, we made a mistake, right? Now when this cell that have this DNA with a mutation, when this cell divide, it still can pass normal DNA to one daughter cells and DNA with a mutation or altered DNA to another daughter cell. Right, so that's a spontaneous mutation. It's just a mistake in DNA replication. Now, spontaneous mutation rate differs between genes. Larger genes usually have higher mutation rates. Each individual has multiple new mutations. So you and me, we have new mutations that neither our parents or our grandparents had. So we have multiple mutations. It just always happen. The rate is rare, but every time DNA is replicated, mistakes happens. Spontaneous mutation also manifests as gonadal mosaicism. Parent has a mutation in some sperm, so we might have so one spontaneous mutation can be gonadal mosaicism. That means that some of the sperm or oocytes carry mutations because of spontaneous process, and other might not, right? So this we're talking about DNA inside the nucleus. Remember, we also have DNA in the mitochondria. So mitochondrial genes mutate at a higher rate than nuclear genes because they can't repair their DNA. Right, so I hope it makes sense. So two types of mutations: spontaneous mutations and mutations that are caused by mutants, some chemicals or cancerogens, right? Or some agents that cause a mutation, mutagen. Is it spontaneous or mutagen? So spontaneous mutation happen really without any reason, just because when DNA is replicated, mistakes happen. And we do have mechanisms to fix those mistakes, but sometimes mechanisms fail to function, and every one of us have a large amount of spontaneous mutation, and those mutations might not even affect our phenotype at all, right? Spontaneous mutation can also happen in sperm and eggs, and then, male produce millions of sperm. Some of the sperm might carry mutations, some might not. The same with females. So we make several oocytes every month, and 1 or 2 oocytes survive and might be fertilized. So some of them might carry mutation, and this is called gonadal mosaicism. In addition, in the mitochondria, we have small amount of DNA that also can carry mutation, and it's more, the rate is higher because there is no repair mechanism in mitochondrial genes. Plus, in some genes, mutations are more likely to occur in regions called hot spots. So short repetitive sequences, like shown over here. So this is short repeating sequence, AAA, TTT. So that's a hot spot, and mutation happens more often inside those spots. Right, and another type of mutation, rather than spontaneous mutation, are induced mutation. They caused by mutagens, and many are also carcinogens and can cause cancer. So example, alkylating agents, and alkaline means the basic solution. So those alkylating agents, they remove a base in DNA. X-ray can break chromosomes. UV radiation can create thymine dimers, and dimers is just a, it's a structure inside the DNA that will interfere with DNA replication and transcription, right? Okay, and this is where we stop today, and we will talk about types of mutations in the second part of this lecture. Okay, so thank you for watching, and I hope it was helpful.